Hi, everybody. Welcome to Q&A with A&V. I am Vincent Racaniello, and joining me tonight from New York, Amy Rosenfeld. Hello, Amy.
Hi, Vincent. How are you today?
I'm very well, thank you. As
Good.
I was told on the train, every day is a good day.
Well, every day that your train doesn't back up because they overshoot the platform, yes.
Hello, everyone. Hello. So far, we have 280 people. I expect we will get in the 600s. And
Why? Because of the whatever it is, the variant,
Omicron. So you have to
Omicron.
listen to TWIV 836, which drops at midnight. And Kathy gives us a pronunciation lesson on Omicron. It's not Omicron, it's Omicron or Omicron.
No, it's
Okay.
Omicron. There was a whole big thing about it in the Times
Oh,
about,
yeah?
yeah.
Okay. I like B.1.1.529. What do you think of that?
It's too many dots. I can't remember.
So just a note to the folks who are here. You know, you guys were chatting before I pushed the live button. And the first question I have is well into that chat. It's from Ada, who says, where I go for my sanity. And I don't know if it's here or somewhere else because that's the only bit.
Oh, I know where it is. It's not here. It's the eighth
It's not
floor of
here?
Saks. No, it's the eighth floor of Saks.
Is
That's where
that where
you
you
go.
go? Okay.
Of course, that's the shoe haven.
Okay, should we dive in, Amy? Amy's got
Sure, because
stripy.
I was going to say, I have to finish my Eliza and I have overlay. So, yeah, we should dive in. I have to leave like 8:45, 8:50 the latest.
Okay, we got stripy tonight, Amy.
Yeah, I got stripes today.
It's good.
Because
Okay.
yesterday they turned off the heat because they claimed that there was a steam problem. It was really, really cold. And this is one of my biggest sweaters, so I can wear like 6 different layers of clothing underneath it, like light
Mm-hmm.
layers of clothing underneath it, and still be able to move my arms. Oh, wait. So somebody says they want to see my shoes, but this reminds me to thank Miriam, right? Who sent me the
Maryam.
Mariam, who sent me the beautiful sneakers that Vincent's going to deliver on Friday. Thank you very much for the lovely Stan Smith Adidas. Can't wait to wear them.
All right, let's start with Elena. I'm sorry. Very apologize for the throat clearing. After this question, we're going to give an overview of Omicron. I have a nice web page that gives you up-to-date information. But let's take this brief overview of the
We have
differences
no
among.
information.
Well, I'll show you what's there. SARS-1, MERS, and SARS-CoV-2. Isn't that what everyone loves about Amy? Vincent says, I'm going to give you the information, and Amy says, we have none. And she's
Well,
actually right.
we don't. I just heard this. So first of all, I heard this epidemiologist at Texas talk on some podcast this morning, and she starts off with, the 30 alterations within spike. Some of them we've already seen, some of them we don't, but absolutely more transmissible. Really?
Oh, she's full of it. This is just
Really?
BS.
She held
Oh,
up the
come
envelope,
on.
and that's what it says.
None of them have been
And
proven yet to be more
exactly. And then
transmissible.
and then they go on about antibody evasion and all this other stuff. And it's like,
Oh boy. So let's do
it's
it. Let's do this, and then we'll get back to Omicron. Differences among SARS-1, MERS, and SARS-CoV-2. So SARS-1
well,
was the first epidemic coronavirus to emerge in 2003. It came from a civet cat via bats in the meat markets in the south of China.
wait a minute.
It
Came
caused
by the bats via the civet cat,
yes,
not the
bats
other way
via
around.
the civets were the intermediary,
Yeah.
and people got infected at live meat markets in Guangzhou. And this virus was hidden from the world by China for quite a few months, and so it spread extensively before anyone knew about it. But fortunately, it was ended after about 8,000 infections. It causes acute respiratory distress and acute lung injury. But the difference between SARS-1 and 2 is that SARS-1, you peaked shedding at the peak of symptoms, which is when you were in the hospital. And over half of the people were in hospitals. So it was relatively easy to contain. There were very few asymptomatic infections, and so not a lot of community spread. And so it went away, and people stopped, except for the coronavirologists, many people stopped thinking about coronaviruses. And then MERS coronavirus, discovered in 2012 in the Arabian Peninsula. This is a bit different because human-to-human transmission is very, very limited. Extremely short chains of infection. As Amy would say, you think a chain of 1 or 2 is actually a chain, Vincent? Exactly. So it goes from a camel to a person. Typically a person who is in bad health, but not always. And then that person can get quite ill, but doesn't really transmit much. It remains in the Arabian Peninsula, and it goes elsewhere by travel. 1 or 2 cases a month, still, but never big outbreaks. Doesn't do human-to-human transmission. And finally, SARS-CoV-2, where we have lots of asymptomatic and mild infections, so very good community spread, but in many people, an age-dependent, largely a serious lung disease like SARS-1. And in all three,
that is much smaller. It's only like 20% or even
right.
less.
That's right.
It's less than that.
And all 3 viruses, the disease in humans is characterized by a hyperinflammatory state. A lot of the disease triggered by the immune response.
A lot of the serious disease.
Serious disease, that's right. The upper tract, mild disease, is just virus reproduction in the nasopharynx. Very much like some of Amy's favorite viruses.
Yes, a lot of my favorite viruses.
Let me show you.
Let's look at this website before we forget.
Where is it? Guys,
I don't know.
Where did it go? Oh, it's not showing up. Safari. Guys, see this? Can you see this website now?
Yeah, I see it. And there's an ORF8. There's an ORF8 alteration.
All right, so let's. This is.
There it says B1.1.1.529. Ooh, goody.
Yeah. So it's currently in 13 countries, 168 sequences.
Here. There's been 1 case here. They should update. It's been 1 case here. Flew in. Did you know it could fly? It got on a plane from South Africa and it flew to California.
It's already widespread. This idea of shutting down South Africa is absurd. It's just ridiculous. The earliest sample you can see is November 9th. And these are some of the defining changes in the genome. You have some deletions up at the 5' non-coding region. Then you have ORF1A.
Yeah, but changes. Who knows where that is? That's not particularly helpful since it's like 2/3 of the genome.
ORF1B, and then we go into spike, where we have 34 amino acid changes in spike, including some of the ones you've come to know and love, like E484A and N501Y.
Yeah, I mean, it's got 8 changes there that are known.
And then we have the E protein, the E glycoprotein, and the other membrane protein.
Nobody knows what it does.
And then we have M, and then nucleocapsid here at the bottom. So these are defining changes. And I will have more to say about the spike changes in a minute. But here's the cumulative sequence count total over time. Okay, that's fine. So obviously this is going places. These are the countries.
Well, that's because it got on a plane and it's going to get in a car. It's not
sequence count per country, 124 in South Africa. This is the sequences and the count per continent. So, Africa here is the highest one. Anything else here? This is basically all the sequences. And there's also another quite nice site here, covariants, where you can see all the variants and you can click on them and get some information on the changes. So here are the defining mutations again. They have a nice table here which shows a list of shared mutations for the 5 variants of concern. So you see the variants of concern across the top here, Omicron in the middle, and you can see these are all shared with other variants. Yeah, these are the spike changes. And then there's some others that are unique to the Omicron right down here, all of them in spike. And what these do is anybody's guess whether they since they are all similar to, they have some changes in common with the other variants of concern, they will most likely affect antibody neutralization. But I just want to point out that B. Nash and Hatsuano a couple of months ago put out a preprint where they put
It's
20
totally
different...
published.
Is it published now?
Yeah, it's a Nature paper.
They put 20 amino acid changes into spike, which they got from resistance mutations to all different monoclonals, polyclonal sera, etc. Some of they selected in the lab, some from people. So they put all the changes they could in, and they put that in a vesicular stomatitis virus background. That virus can still be neutralized by human sera from people who have recovered from infection and then gotten one booster, one vaccine dose. And they say in the paper, this says that any variant that comes up in the future should be taken care of by vaccines. And so to make a big deal out of this is kind of absurd when we already know that kind of virus can be neutralized by sera. It's not fully neutralized, but it can be neutralized. And don't forget T cells. The other point here is that the T cell epitopes in Omicron have not changed. And so the T cells will still save you from severe disease and death. That's the overview. And so those of you who are asked for our opinions, I don't think there's any evidence to suggest that it's more transmissible or more virulent, but no variant has been definitively shown to be more transmissible or virulent in this virologist's opinion, and also the one sitting next to me on my left.
Oh, is that me?
It would be you. So there we go, and you can follow up, and we will take...
I think that so there's only so many changes that can occur before it can no longer use ACE2. So there is not an infinite number of changes. It will eventually... So to me, this is like the paper that we did on measles. Mm-hmm. Remember that? Where actually the measles and polio, they undergo antigenic drift until they can no longer use the receptor.
Correct.
And since the immune dominant epitopes are the ones that block receptor binding, that once you no longer are able to use the receptor, you're resistant to the antibodies. But it doesn't matter because you're not able to infect the cell to begin with. So I think that unfortunately, people like the epidemiologist at Texas that I heard this afternoon or this morning doesn't really know enough virology and basic science to understand that and communicate that in a way that doesn't monger fear.
Fearmonger. As DGB says, can you really predict transmissibility based on where mutations are?
Well, of course, doesn't he know anything? God!
Only if you know which changes affect transmissibility. So, for example,
you don't even need to know that. You just need to talk to the envelope.
Oh, the envelope.
Everybody knows that.
I have an envelope tonight, Amy. I'm going to put it up and say
that's blue. Is that the spike envelope or is that the membrane envelope? Because last time the spike envelope was yellow.
I don't know. This is my AAA renewal, so it's blue.
I see.
I don't drive anywhere anymore. Maybe I don't need AAA. But folks,
you need AAA. Your car's going to not work because you don't drive anymore. Therefore, you're going to need AAA.
There's only one situation where we know some of the amino acid changes in a glycoprotein that go with transmissibility, and that is avian H5N1 influenza virus in ferrets in a very controlled environment in the lab, in cages where the number of ferrets don't change, their interactions don't change, and there you can measure transmissibility. And no matter what...
you can measure transmissibility between ferrets, not
between ferrets, humans. Absolutely. Anyone tells you anything else, they're wrong, because you can't do that experiment in people. So to say this lady in Texas said it, this epidemiologist, I should say, is simply wrong, but that's what the press is picking up. As Amy would say, scientists are the ones who contaminate the press.
Yeah, they do. And she was on, she's contaminating, and he bought into it. The
And
interviewer bought into it,
and
hook, line, and sinker.
that's why they don't.
And then he says at the end of it, like, next week he's going to have this great scientist that he doesn't want to tell anybody who it is on Wednesday. And I'm like sitting there going, really? You're going to have a great scientist? I'm still waiting for you to have, yeah.
So the reason that they don't have me and Amy on these news shows is that we'll say, don't worry, it's not an issue. This is SARS-CoV-2, folks. Just think if we hadn't done any.
I don't want to go on these news shows. I'm too busy working on EV68 and enteroviruses.
Just think if no one had done any sequencing, and we would have a sort of scenario where we would have waves of infection. We'd get people vaccinated. They would go down. They would go up and down. And eventually it would stop, and nobody would be freaking out.
Wait a minute. Who said don't do any sequencing?
Well, you said don't sequence vaccinated people.
Oh, yeah, no, I said don't do any sequencing unless this is an experiment, unless the results are being used as an experiment to understand adaptation to a new host. Don't go out sequencing people who come in and test positive. That's what I said. Go back way back, all the way back until like April. Way back into the archives. And you will hear me say, unless this is a scientific experiment, there is no need to be sequenced.
I agree, but you would be crucified by the sequence jockeys who like to analyze the sequences. But I'm saying that the outbreak, the pandemic, would be no different if we had zero sequence and if we just made these vaccines and vaccinated people.
It's going to take 5 years. I'm the person who's going to take 5 years, and we're already at the end of year 2. And we've had more deaths this year than we had last year, and we have an increased number of cases. People are doomed to repeat 1918.
All right, so those of you who wanted to know our opinions, that's what we think, but we can take more questions later.
I can't take any more.
Thank you, Vanity, for moderating. I thanked everyone else, and I didn't see you.
Oh, Vanity Animal Party would be a great one. Yeah, she's making my beautiful painting of EVD68, and then she sent me these really pretty photos of her viral snowflakes, and she says to me, do I want glitter on them? Like, seriously, who doesn't like glitter?
Anyway, Vanity was in the incubator this morning for Livestream Virology, so thank you, Vanity. It was a pleasure to meet you. Okay, Westfield 90 wants to know, he got his booster. If Pfizer comes up with an Omicron vax, does that mean your booster's useless? They will not. There's no reason to have a variant-specific vaccine because when you are immunized with the original spike, your body will make antibodies against all the variants that could ever come. That's how good your immune system is. It's called, well, I'm not going to tell you what it's called.
It's hypersomatic antigen diversity.
Somatic hypermutation.
Yeah, whatever.
You just have to make sure you give the vaccines time to do that, which means...
No, we don't need to do that.
3 to 4 weeks.
No.
It's not
good.
We
don't need to do that. Because that would mean that, again, Amy was right. So that we can't do that.
So there's no need to make...
an entire pandemic of where Amy was right.
There's no need to make variant-specific boosters. The original ancestral is fine, and... especially if you've infected and gotten one of those, you're great. Okay. It's been said that South Africa has made significant contributions to genome sequencing and the work done in the openness. From which indices, databases could one prove this? Well, the databases that share the SARS-CoV-2 data, GISAID and nextstep.org, they all have a lot of contributions from South Africa. But apparently since the travel ban, now they can't get reagents to do their sequencing. Kind of silly, isn't it? If Amy were head of CDC messaging, how would things in the U.S. be different now? Amy doesn't want to be head of CDC, right?
No.
I don't think you would get her to do it. So but
no.
It would be very honest. I'm telling you, it wouldn't say one thing one week and change the next week.
Yeah.
it would be so blunt that Biden would call her up and say, Amy, you need to tone this down.
No, I'm smart enough not to want any of these offices. I think, yeah, I'm smart enough not to want any of these offices. What do you think about that? And you're at 876 people. So I don't know how you got, I bet you by the time you're done in 2 hours, you'll have 1,000, but I won't.
This is a record.
I won't have 1,000 because
they come.
I'm leaving at 8:45, 8:50 to finish my experiments.
They come for you, Amy.
They don't come for me. They just send shoes and wine.
It's not bad.
No, but they stopped sending wine, and somebody said I should drink heavily. So I'm like, if you don't send wine, how am I going to drink heavily?
All right. I'm confused with the vaccine escape of Omicron. Don't we already get sick? The vaccinated, I mean, thank you for everything. Yes, the vaccinated can get mild disease, for sure. Why is that? Because antibody levels contract after vaccination. And so then when you are exposed to the virus, it reproduces for a few days in your nasopharynx until your memory kicks in. And in those few days, you can have an upper respiratory tract infection and symptoms. Now, some people will die because the vaccines are not 100% preventing hospitalization and death. They're only in the 90s. And then if you take, let's say it's 8%. 8% of a lot of people is a lot of people dying. So it all makes perfect sense.
Yeah, did you know I'm as subtle as a brick?
That's why we love you.
I don't understand these people. Like, if you have nothing nice to say, don't push send. Like, it's just not that hard.
Leave me alone, please.
Not that hard.
But the thing that is good about Amy is that she tells you what she is thinking. You never wonder. So here's the thing. In New York, people tell you what they think. In California, they say you're your friend, but they actually hate you. So they can't be honest. That's the way I've learned the world works. And Amy is New York.
It's true.
Amy is
true.
New York.
And the thing is I tell you what I'm thinking, and I tell you how I got there, right? And I give you the option of, if I'm wrong, to correct me.
Yeah. So in response to Eleanor, who asked what would be the difference if Amy were ahead, Westfield 90 says it would be a different pandemic, much information and less stress. Although probably Amy would be stressed, right?
I'm always stressed. When do you think I'm not stressed? Huh? I'm waiting. Even this waiting period is stressful.
I have to think. I have to calculate. You're always
Even
stressed.
This waiting period is
Look,
stressful.
as I said yesterday, even if you had 10 grants and 50 people, you would still be stressed about something. You're never going to be non-stressed. It's genetic, okay?
Well, 50 people automatically is going to stress me out. I can't keep track of 50.
Okay, Carlos wants to know, I hear you saying you probably shouldn't test vaccinated individuals. Is this because we don't know if they transmit? I assume you would only want to test if they had symptoms. Thanks. So what is your logic, Amy?
No, I don't want to test at all.
You don't want to test anybody today?
No. I don't want to test anybody who's vaccinated.
I thought last week it was just vaccinated people.
No. Right, that's what I'm saying. Is I don't want to test anybody who has been vaccinated. Anybody who has not been vaccinated is a separate issue.
All right, let me give you a scenario. Let me give you a scenario. So we have a person with diabetes, and they're 80 years old, and they've gotten 3 doses of vaccine, and then they begin to have respiratory symptoms. Don't you want to know if they have COVID so you can treat them with monoclonal antibodies?
Well, define symptoms.
They have.
You would treat them. Would you test them if they had flu? If you thought that they had flu? So if I'm vaccinated and I'm 80 years old against flu and I have flu symptom and I have respiratory symptoms, do you test them, or do you just give them flu mist or whatever the therapy is for flu?
Many places will just give, if it's the winter in the Northeast, for example, they'll give you flu mist without even testing because you'll most likely have flu. Although now the metric has changed that we have a second virus that can cause severe respiratory illness.
So
Anyway, I'm
if
saying in some
So
cases
what's
you
the
might,
difference? What's the difference?
but we don't have an antiviral to give. We have
Well, you
monoclonals.
will. I mean, it was just approved. Merck's was barely approved, but it was approved, and probably by
Molnupiravir,
yeah.
Thorus hammer. You could just give that, yeah, maybe. But Daniel said they want to. Was it approved for everyone or just unvaccinated people? Because
I
Daniel
think
said they were.
everyone.
Daniel said Pfizer only wanted it. Merck only wanted it for. Oh, maybe that was the Pfizer protease, only for unvaccinated people. But anyway,
Whatever.
yes. That's all I'm saying is that in some cases you may want to test because you have an at-risk
But the majority
population.
of the majority, you've always had a mixed population, so that's not a good rationale because you've always had a mixed population.
Yeah.
So what you're telling me is up until this point we didn't really care if the old diabetics died. Is that what you're telling me?
No, I'm not saying you treat them with antiviral, with flu antivirals for sure. Yeah.
But you didn't test them.
No, because at the time it was only flu in the winter that was going to
No,
make
there's
them
RSV.
sick.
There's RSV.
Not as frequent as flu in older people. It does, but not as frequent as flu in older people.
Of course it is. That's why they're making a vaccine. And one of
the vaccine's for kids.
One of the target groups is for older people, especially
Amy,
people in their
all I know is that before COVID, flu was the big worry, and you would get those flu-like symptoms, and you didn't. Many doctors didn't test; they would just give Tamiflu, for example. But we don't have that option, and a monoclonal intravenous is not. Yeah, just go get a monoclonal. It's
it should
deal.
be.
Okay,
it should be.
That's my position. Here's a question: How different would the spike need to be to evade protection? So,
basically, not be able to use. It would basically not be able to bind ACE2.
So the polymutant that B. Nash and Hatziano made, they made 20 amino acid changes in spike, and by the way, Omicron doesn't have all of those; it has a fraction of them, and even that could be neutralized by convalescent sera. But the key here is that the T cell epitopes are not changed, so the T cells will still recognize virus-infected cells. So it wouldn't evade current vaccines at any point because the T cells do not get evaded ever.
Right, but it's still does it bind ACE2 with equal efficiency?
Which the polymutant?
Yeah.
They didn't look at that, but it is attenuated. It's in a pseudovirus, and it has a log less reproduction. It has a fitness cost associated with it, and that's the other thing. If you make enough changes in spike, that virus will never propagate. It'll never compete. And
that's what I'm saying. That's what I'm saying.
And so we don't know whether Omicron can compete yet. It's too early to tell.
What I'm saying is that in order to completely evade immune evasion and not be neutralized, the virus can no longer evade antibody immunity. The virus can no longer use ACE2. So now if it has not adapted to another protein, it does not matter because it can't infect you.
Right. So I agree, but I don't think it would ever get to that point because T cells would still be able to recognize
but clearly cells. Measles gets to that point. That's what the paper showed, that there was seven amino acids that could be changed in the RBD, and it could still use SLAM and CD46. And then when you make that eighth, it could no longer use either one, and therefore it didn't make a difference.
Yeah, that was made in the lab, but I'm just saying that would never arise in people. They did that in the lab because they wanted to make a vector for
I know what they did
therapy,
it in the lab for, but I'm just saying.
But in nature, that wouldn't arise because as soon as you lose receptor binding activity, you're dead. You're dead in the water.
Right. That's why there's no such thing as soluble resistant PVR polio.
Yeah. So this is a comment that you could agree with probably. I'm growing increasingly annoyed with Topol and other non-virologists making definitive statements about stuff they know little about. Hey, welcome to the pandemic.
Yeah, you're a little late.
It's what's been happening all along, but we have our little corner of the world here, and you guys found it. So good for you. Thank you, Galaxy, for your contribution. I don't think recovering from MRSA would help you with COVID. No. Maybe he thought you had a good immune system, which could, but specifically MRSA, no. But having a great immune system, but you really don't know that. So I'm afraid you can't conclude anything from that. What podcasts do we listen to? Well, since I don't drive anymore, I stopped listening to podcasts. And Amy doesn't, as far as I know. She listens to TWIV, right?
Yeah.
Because this is how it works, I'll tell you. Saturday at midnight, Daniel's TWIV drops. And at 9 a.m., I start to get texts from Amy saying, why did you say that? That's wrong.
9 a.m.? Really? I'm that late?
But before I stopped driving, Ellen, I used to listen to a lot of different podcasts, like tech podcasts, plant podcasts, some history podcasts, but no more.
You really
because
liked Pod Save America.
I liked Pod Save America for a while, but then they got too hysterical. But now I take the train and I do work on the train. I read papers for pods, I make show notes and so forth. It's a really good use of time. So we were at the highest number of participants we've ever had, and this is just amazing because it's a variant pushing this.
Well, then you shouldn't knock the variant. We should embrace the variant. It's making you wealthy and famous. So
I'm not getting wealthy. No.
It's good to have a variant then.
I'm not getting wealthy. I don't get anything. It goes to micro
you're getting
TV.
wealthy in the number of viewers, right?
Yeah.
And more words go around, so it's good.
Look at this. I got to watch
this. A new
this.
variant.
I get to watch this on my media room TV because nothing else is on. A&V on the 70-inch. Cool. Do we look good? I know Amy does. I don't look good.
70-inch? They make
70-inch TV.
What wall can fit a 70-inch TV?
What do you have? How many inch TV do you have?
I don't know. You bought it with me. What do I have?
No, I just carried it to your house. You bought it and you said, I need to get this to my house. So I put it in the back of my car.
No, you helped me pick it out.
I did? Online, you mean? Okay, I don't remember.
Online. I don't know what we bought.
So if the Biden administration asked you or Amy to join their advisory team, would you? I would, yeah, sure. Would you?
Sure.
If Amy were on it, though, I wouldn't join. I don't need any more. I'm just kidding. Of course I would. Of course I would join. Because then we could talk behind their backs, right?
Why do I need to talk behind their backs? I could say what I need to say and then move on.
The meeting would be over and you would text me and say, can you believe that stuff?
Well, actually, I would just have to send you a picture of my calf because you would spend the entire time kicking me underneath the table saying, don't say that, don't say that, don't say that. It's kind of like the story of Andrew and our friend Elliot, whose wife didn't want him to eat a steak, but he ran a fur store. And Andrew just kept kicking me saying, don't say anything, Amy, don't say it, because I can see it.
This is why Zoom has
the hypocrisy of it all. And look, you're at 1,011 people.
Congratulations. We hit 1,000. Andrew from New Zealand, you predicted it months ago, but here we are. I would say never because I didn't think another variant would do this. But this is why Zoom has a chat feature. The other day, Amy and I and another person were on a call, and she was getting a little out of hand. So I had to chat her, calm down.
And then I yelled at you.
And then you said, stop yelling at me. And I said, I'm not yelling at you. I want you to behave. That's all. It's very difficult.
Yeah, it's very hard being you. My heart bleeds for you.
All right. I heard from a papers abstract.
I appreciate my humor and my personality.
All right. Listen. Let's move on, Amy. Incorporation of short host RNA sequences during genome replication is possible for SARS-2. Yeah, sure.
What?
So I'm going to tell you, Amy, remember the story where poliovirus picks up a piece of ribosomal RNA?
Yeah, very similar.
Yeah, the polymerase, it's copying the viral genome, and it could collide into another RNA that's around because the cytoplasm is very crowded. So this is completely possible for SARS-CoV-2.
Yeah,
It
but
just
it's fine. Who
hasn't been
cares?
shown yet.
Who
Hasn't been
cares?
shown yet. Let's see. So we have 1,018 listeners. Maybe we could get like 900 thumbs up. Or am I asking for too much?
I don't know. I don't have a button, nor do I keep track. That's your shtick.
I'm from New Zealand, where currently we use Pfizer and AstraZeneca. A lot of my friends are suddenly saying they want Moderna. No, Pfizer and Moderna, they're fine. There's some studies that say there's a slight difference, but it's not meaningful. Any single study is simply not meaningful. So you're fine with Pfizer. Absolutely.
I got Pfizer. I'm fine.
Okay, I frequently cross from Canada into the U.S. I'm very sorry. Which requires negative PCR in addition to distancing, masking. Does getting boosted reduce my odds of positive PCR? Probably not. I don't think it would. I think, as Amy would say, if you get exposed, you're going to have some RNA in your nose, even if it's not reproducing.
Yeah,
You get
exactly.
a positive PCR.
I'm not clear how being vaccinated protects you from exposure. It's like I put on a lot of clothes, and when I go outside in the summer, I'm still exposed to the sun, so I don't really understand. I don't get it. I'm not that smart, though, you know. So
Neither am I. I just
that's
work
why I don't
hard.
get it.
What's all this fuss about the notorious RBD? So Truth or Dare often makes "what's all this fuss about" jokes, which you like to do, right? What's all this? You like
Gelda?
Gilda, right?
I don't know. I never really watched her. She was before my time.
Yeah, RBG. She said we can disagree without being disagreeable.
Yeah, I think that's fine. It's good.
It could, if a virus can end up with host proteins on its surface, could this impact a person's ability to transmit, as now virus particles have more antigen to which people may have previous exposure? No, they're self, so they're not going to be recognized as foreign. You don't make antibodies against your own proteins normally, although some people who have autoimmune diseases do. Your sweater is loved.
Oh, thanks.
Ellen is a fan of yours, clearly.
Yeah, she's got a Star Wars t-shirt on. You never saw Star Wars either.
What do you mean?
I never saw the movies. I've never seen any Star Wars. Move on.
Peter says, in my humble opinion, I think the most important conversation is Omicron and whether it is the variant that will show this is nature's way of progressing versus man's ability to master science. Okay. You're entitled to
I don't really understand it.
Well, he's saying we should be talking about Omicron.
Omicron?
This is what everyone wants. The 1,000 people, or I would say, I don't know, probably 400 of them are here just to know about Omicron. And there's nothing to worry about.
There's nothing to know. You have to, there's nothing to know. It's going to take you a month to do any experiment in the laboratory.
But
so
I say that no matter what's found,
right.
The vaccines will protect you because the T-cell epitopes have not changed. How many times can I say that? The T-cell epitopes have not changed. T-cells protect you against severe disease, hospitalization, and death.
It's not even clear to me that just because it has changes that it's A, more transmissible, which clearly you can't deduce ever, or B, it's going to correlate with increased disease severity. So they've been saying like, oh, Delta, more transmissible. Well, it didn't increase the disease severity. So what do I care? So it's changes. Okay.
Yes, I agree. What would it do to your behavior? Would you just stay home? Maybe some people would if they didn't have to go out. I don't think that really matters all that much. If a triple vaccinated individual tests positive but is asymptomatic, should they be rushing to get monoclonals? You should go to your doctor, because if your doctor was Daniel and you had a comorbidity, probably Daniel would give you monoclonals. Yeah. So you have to consult your doc. I think it's the high-risk individuals that we worry about. Although I have to say, If you
You
don't
don't give
have,
them monoclonals for flu, and there's high risk individuals all the time.
well, that's not my decision, Amy. I forgot what I was going to say. All right, it'll come back. There are people without obvious comorbidities who clearly have something, because then they do get severe disease, and we don't understand why.
Well, I'm not walking around with a sign above my head that says I'm immune compromised against measles. Do you see a sign? It's blinking.
Yeah, I was reinfected between my first 2 shots. I am finally overcoming long-haul COVID. Do I need to risk being exposed to others for a 3rd shot? I don't understand your question.
So she thinks like the people who are going to give her the 3rd shot are going to expose her to virus. But
I don't think
I
that's
mean, she must have like a huge, like according to all the papers, then she has a huge immune response. So she's completely protected because she's been vaccinated and she's been infected.
Mm-hmm.
So I don't get it. So there's no problem.
I think you're good overjoyed.
Very much.
Got 2nd dose 5 months ago. Have to speak indoors close to vaccinated people without any masks. Should I get a booster? I'm not sure it's going to reduce your chance of spreading virus. You're vaccinated. The people are vaccinated.
Well, why would you wear a mask? I mean, why
They're not
would
wearing
you?
masks. You can't.
Right. So she's vaccinated. The people are vaccinated. So who's going to get infected? And who's going? There's like no chance. There's a very low chance of severe disease in that case, right? And even of getting infected, because most of the infections would result in aborted virus reproductive cycles.
It depends how old you are. Also, if you have any comorbidities, right? That could play a role. If you are older, you have comorbidities, maybe you should do that. Jeff wants to know if I'm bringing you on my Nebraska trip. I hear she's always right, and I need access to the guru. No, she's not. She has to stay behind and work.
She has to stay behind and work. I was not invited.
You've been to Nebraska, haven't you? Didn't you go there, or was that South Dakota?
I was South Dakota.
Okay.
It's okay.
No,
I've applied to no, I didn't even apply there. No, he goes when you write him, you are inviting him to do a TWIV and talk about research or whatever you want him to talk about. You're not inviting me. Why would I go?
Yeah, no, they invite me, so they pay my way. So Amy would have to go on her own. She gets invited elsewhere.
I don't get invited anywhere, but I'm fine not being invited.
You went on 12 job interviews in the past 5 years, right?
Yeah.
Geert van den Bosch makes wild claims about safety of vaccines. Meanwhile, he has his own vaccine in development based on NK cells. Of course. That's what it's all about. They want you to buy their product. Does his vaccine have any potential? Well, you could make a vaccine that's based on NK cells, but I'm not sure how much of a... NK cells are cells that kill virus-infected cells by virtue of them lacking MHC on the surface. I'm just not sure how you would target a vaccine to them and whether Geert knows anything about that. So I would be highly skeptical. But I await the first paper. That's the way we do it in science.
All right, 2 more, and because it's 8:43. 2 more.
Oh my gosh, how fast the time went, Amy.
Yeah, it's always fast.
Does respiratory syncytial virus halt mitosis in telophase? And if so, how? I'm not aware of that. Are you?
No.
Inhibiting telophase? No, not that I'm aware of.
Not aware of it.
Is lifting patents on vaccines useful? Where are the 3rd jabs coming from? Could you have a guest on to talk about global vaccine manufacturing distribution? Yeah, I had wanted some time ago to have a supply chain person on. Yeah.
But it's not about it's not about lifting the patents. The patents have nothing to do with whether or not you have the manufacturing capacity and how fast you can build the factory to, especially for the mRNA, you need like very specific robots and stuff, and it has to be very sterile because otherwise the RNA degrades and stuff. It's not that easy.
Amy,
It's
can I
not
ask
about
you,
the patents.
who was the supply chain person? Was that the podcasting guy? What's his name? The guy who does podcasts you're always telling me about. He was in the
So
Obama
Andy
administration.
Slavitt.
Is he a supply chain guy?
He had a supply chain guy. He had a global person too from England, and he had both CEOs from Moderna and Pfizer, and they all say it's not about the patent, it's about the fact that there's no manufacturing capacity. And you can't just convert a factory that made like flu vaccines into this kind of vaccine. Like they have all the like the robots sense. And the robots are not the same and everything, so you need different things. But and there have been a lot of other commentaries on it about it's not like, even when Merck and Pfizer removed the patent for their drugs, it turns out that it's not that they're still predicting like it's going to take like 2 or 3 years for the drugs to go to like Africa because they or Far East Asia or something, like around Vietnam, because they don't have the manufacturing capacity. They don't have the factories.
So I asked you about Slavitz because I'd like to get someone on to talk about supply chain issues with drugs and vaccines. And do you think he would be a person or someone else?
Andy, no. But the person who knows about it would be, yeah. I think the commentator would be. But not Andy. Andy's a mediator.
Okay.
A moderator. And the other thing is crap, I was going to forget something. It's not just this. So now we have supply chain problems that are driving the inflation, and like you can't really even get, like, we're still behind in getting tissue culture plates and crap. Like we literally, because I paid a P card, we got an order that I filled last week from Fisher. Do you know how long ago I placed that order?
8 months.
Yeah, February.
That's surprising.
Because the Fisher site, they automatically give you a list of your invoices for the first 6 months, for the 6 months from today. And I couldn't find it, and I couldn't find it, and I couldn't find it. And finally I realized that on the dropout menu it said you could do more than 12 months, and I had to push the button that said more than 12 months.
How often does a bright mind in academia not make it because they stink at the bench?
Really, about a huge amount of the time.
Would you say over 50%?
Yes.
Yeah, many people just don't have good hands.
If you can't you can't even, they don't even get to the academic part. They fail at graduate school.
Amy, what do you think of the idea that the number of
I have to go.
unvaccinated people has nothing to do with the generation of variants?
What do you mean that the unvaccinated people have nothing to do with the generation of the variant?
So the idea that the more unvaccinated, the more variants, some people are saying that's not true. What do you think?
Where did they think that the variants came from?
I would argue that.
It's like when people say to me, well, there's not been 700,000 deaths in the United States from COVID, which is a complete underestimation, because if you listen to Daniel, you can't always put that they died of COVID on their death certificate. So it's totally an under representation. But even let's say it's right. Where did those people go? To the spaceship with Jeff Bezos? It's got to be a really big clown car spaceship then, right? The clown car,
the various in vaccinated people, there's not a great deal of reproduction, so variant generation is low. Variants get produced in unvaccinated people. There's no question about that.
Yeah, the more you replicate, the more variants you're going to make, because if you make alteration every cycle, right? So if you replicate 1,000 viruses, you have 1,000 viruses that have different alterations. If I replicate two, I have two viruses that each one has a different alteration. I don't
Yeah,
understand
that's
that.
it. Pretty straightforward, right?
Yeah, exactly. Do the math. I don't really understand
Well,
what the
that's
issue
why they
is.
asked you. They asked you to learn that.
Well, they
Okay.
didn't ask me. They asked you.
Well, no, this is for both of us. It's Q&A with A&V. Are you done?
Yeah, I
Did you
gotta go. I
you
gotta go
sure?
do. Yeah, I gotta do my podcast saying my Eliza. Are
All
you
right,
gonna
all
come
right.
too?
Thank you. No, I thank you for joining us. I just know that someone is saying later you should go on Rogan. That would be a gas.
Why do I need to go on Rogan? I don't even know who he. I don't even listen to him.
Yeah, but
But what?
the conversation between the two of you would be so funny.
So
It would
it's
be.
like a comedy show?
It could be a comedy show. Yeah. Well, sometimes he's funny. I don't know. I don't watch him.
I
So
don't watch him, but like I'm the person who got yelled at when I said like that Lex was a hypocrite or a fraud, whatever,
well,
bullshitter,
this
or
is
whatever
why.
I
Look,
said.
Amy speaks her mind, and whether you agree with it or not, that's a good quality. All right, Amy. Thank you again for
All
joining
right,
us.
I'll talk to you tomorrow.
Yeah. Take care. Good night.
Bye.
All right. After flagging there's no N, Omicron. So Omni is what Biden said. That's not right. Omicron is the travel isolation of South Africa warranted? Not at all. The virus is already out, right? The horse has left the barn, if you want to be cliched. There is no reason. All it does is put undue hardship on South Africa. And so if I were in charge, it wouldn't happen. But I'm not. But maybe you guys are lucky. CDC states that close contact is within 6 feet. That's correct. But they make no distinction between indoor and outdoor. Outdoor is safer, of course, because of the dilution factor, right? Especially if there's some breeze, it's moving the respiratory droplets around. And so refreshes the air, and it's definitely better. Exactly how much, I couldn't put a number on it. I would be making it up. Dr. Hotez says it's similar to alpha. Well, I don't think you should say that, Dr. Hotez, until you do some studies on the variant. Don't just look at the changes. Here's why. We saw in this, let's go back to the table, okay? Let's see. This is common changes. So here's alpha, and here's Omicron. So we have a change in common in spike right there. That's 1, that's 2, that's 3. And then there is N501Y, D614G is in everybody, so that doesn't really count. Then we have this one, okay? However, there are also changes in common with other variants. And so you don't know how that shakes out. So I don't think that's something worth saying it's similar to alpha. I don't mean to criticize Peter, but that's not something I would say. Why would you say that? I'm fully vaccinated. Is it safe to go to dinner with friends? Some are not vaccinated. If you're fully vaccinated, in my opinion, the worst you're going to get is a mild illness. However, if you're protected 92% against severe disease, there's still some chance that you would get severe disease. So it's up to you. If you think less than 10% chance is worth it, by all means go. Those are the numbers. I mean, I go to dinner occasionally in New York, but there they require everyone to be vaccinated. You can't get in the door. So, and, I'm on the train. Not everyone's vaccinated, but they're all wearing masks. So that's what we're doing right now to protect everybody. Merck's antiviral pill is going through the works. It should be out, as Daniel says, before the end of the year. Yeah, Vanity got to see the sneakers. They're very nice, yeah, and it's really funny. Yeah, Amy was fired up, and I'm amazed at how fast the time went, right? Would getting a blood test a week after getting an mRNA booster negatively affect one's immune response to the vaccine? Absolutely not. No effect whatsoever. Would a J&J second shot be beneficial due to its T and B cell advantage in conjunction with an mRNA? So you've got one shot of J&J, by all means get a second. Absolutely. In fact, that's what's being recommended. If you have only one J&J, get a second one at least 2 months after the first one. Now, you're asking whether adding an mRNA would help. That we don't know because it hasn't been looked at carefully. It could, but I couldn't tell you that it would. In which group of 10 plates do you get more variability of the quasi-species, infecting 10 plates simultaneously with the exact virus versus sequentially? Well, in each plate, you're going to get the same quasi-species out, assuming that the conditions are all the same. And if you passage the virus, it depends on how much virus you're using. If you use a lot of virus, then the quasi-species will be maintained. I'm fearful of being sick. I understand that. I totally understand that. And I know that from your visits here over the last year, you've had a lot of difficulties, and I'm sorry about that. All I can say is this will end, and you're not going to have to be sick anymore. I'm quite sure about that. But I do appreciate that you come and listen and try to make sense of all of this. I think that's important. What do you make of all the BS going around the virus and how it's become so politicized and polarized? Well, because people see they can take advantage of it like anything else. Any other situation that gives one person an edge over another, they take advantage of it. I do think, though, that people's health shouldn't be messed with. It shouldn't be made a political endgame. It's really unfortunate that it has been. So I'm not I think it's awful. But, vaccines are the same. They're manipulated for political means, and they shouldn't be. They should just be given to people to make them healthy. I think that is one of the jobs of governments, to make sure people are healthy as well as educated. And you're not doing it if you're playing games with vaccines and masks and so forth. Can you talk a little bit about why the COVID vaccines can slow down an infection, but in many cases unable to eliminate the virus? All right, so I think we just did, but I'll repeat it again. So when you're immunized, or if you recover from infection, you have antibodies and T cells. They very gradually contract. The levels contract so that maybe in 6 to 8 months, now the levels in your blood of antibodies and T cells are very low. Not zero, but they're low. But what you do have is memory cells. And so then when you are exposed to virus, the virus can reproduce in your nasopharyngeal cavity because you don't have much antibody to stop it. But in 2 days or so, the memory cells get activated, they start making antibodies, and that will reduce the overall amount of virus made, the overall damage to you. And so in those first few days, you can have a mild cold. Or maybe not so mild, maybe a little more severe because you have a different immune system from others. So that's why, because more than 6 months out after a vaccine, after an infection, you don't have high enough levels to really prevent infection. No vaccine works that way. Very few do. And certainly if you've recovered, it doesn't work that way either. You get a little bit of an infection. These vaccines are no different. And we've changed the bar in this story. We started out saying, hey, we'll take any vaccine better than 50%. And then we got 90-some% against all COVID. And then when that started to drop, we freaked out, but we failed to tell you that we're freaking out over all COVID, not just serious COVID. The serious COVID remained high. And so it's unfortunate, I think. Do I think Omicron came from someone with a chronic infection? or zoonotic spillback. Yes. So you've been following the med Twitter, as Jeff would say. These are all the discussions going on med Twitter. Personally, I like the TWIV discussions. That's where I have mine. But those are both possibilities. Sure, it could be. And in fact, we know that in people with poor immune systems and they get infected, they can harbor virus for a long time. The paper was published on that a long time ago. And the kinds of mutations that arise in those people are also found in variants. So it's entirely plausible that these different variants arose in people with long-term infections, but we can't prove it. We don't have a direct connection. Zoonotic spillback, this is Christian Andersen's idea. He thinks maybe the virus went into animals and it changed and now it's popping back into people. Well, we would never know because we don't do enough surveillance of animals. We now know that deer are heavily infected, and in the state of Iowa, that story is very interesting. It's in preprint, and we're going to have the senior author of that story on TWIV shortly. We're arranging a date with him. That'll be interesting. But other wildlife, we're not looking at it enough. Or maybe we are and we haven't heard yet. So there's just no way to prove either scenario. But I think they're both possible. Or that it's just moving through the population. I have to point out that a couple of weeks ago on TWIV, we did a paper, it was in PLOS Pathogens, where they selected SARS-CoV-2 variants resistant to remdesivir. And they did this by passaging the virus in cells in the presence of remdesivir. And they got viruses somewhat resistant. But at the same time, they said, why don't we sequence the spike and see what's happening there? And guess what? They found the same bloody amino acid changes that are found in the variants of concern, like N501Y. So the moral of the story is that you don't need to have immune pressure to select out. They just pop up when the virus is passing. So my view, one view, another view, maybe a third view, is that as the virus passes among people, it just sustains changes. And I don't think don't view it as a progression to something better. It's not, because beta was actually better at evading antibody than is delta. So different properties go up and down as these various variants. And all that's selected for is fitness. The most fit virus predominates. And what are the changes that mediate that are very difficult to tease out. You'll see we had a discussion about that on TWIV just yesterday. These are good questions, folks. Very good questions. Any studies on genetic predisposition markers present in people who have not caught COVID? If so, would they correlate with the subject's lower predisposition to catch other coronas? So I'm not aware of any. The main ones are the studies in people who get serious COVID. We want to know what's going on there. But I agree, equally interesting is what makes people recalcitrant to, not the right word, not able to have serious COVID. And I'm not aware of any studies that address that, but would really be a good idea. When MHC presents antigen on the cell's surface after mRNA vaccination, is the antigen's orientation random? So it is not the whole spike. When it's presented in MHC, it's just the peptide, of 8, 10 amino acids. I don't remember the exact length, but it's pretty short amino acids. And so, and the MHC, the top looks like a bun, a hot dog bun, and the peptide looks like a hot dog laying in it. So there's no upside down spike because there's no full spike there. How long does it take to grow enough coronavirus to do tests? Depends what kind of test, but you can grow coronavirus in a few days. It's quite easy to grow in cells in the laboratory. If you want 100,000 liters worth, then it would take you a couple of weeks probably to build up to that level and to do the infection and so forth. I sense annoyance with Omicron proclamations in the absence of good data. Well, frankly, Tom, it's been the case throughout this pandemic, and we still don't have much good data on Delta. We have now some lab experiments coming out which are really needed to address the issue of why are these viruses more fit. That's the bottom line, and I know half the people here don't buy it from me because they say other experts are saying otherwise, but that's just wrong. They haven't looked at it the correct way, the virological way. So we will have to start studies on Omicron to see, well, first of all, we'll see if it goes anywhere. It seems to be moving around the world. We'll see if it displaces other variants. We'll know that in the next couple of weeks. And if it does, that means it's more fit, and then we have to find out why. These are all interesting questions that take a long time to sort out. Why does obesity make you at greater risk? So obesity is essentially an inflammatory disease. And so severe COVID is an inflammatory disease. And so you're at increased risk for a more severe inflammatory disease. That is how I view it. But it is not how everyone views it. What do I think of Dr. Hotez's vaccine? I'm not sure I know if it's the inactivated vaccine. I don't think it's the inactivated. I think he's doing a protein-based vaccine. I don't know. I haven't seen the numbers. We just reviewed the numbers for the inactivated Covaxin, which look really good. 2 doses. Do you think Omicron could be what we need? A variant more infectious, less severe disease, so more like other coronas? Well, there's zero evidence that that's the case, and you cannot conclude that from 168 observations all over the world. I do think that, there's a prevailing idea, I would say, that viruses over time become better adapted to their host and cause less severe disease. But I think that's a long period of time. I don't think it's in 2 years, so I doubt it. It's a good idea, but I don't think we're there yet because there are too many people who have no immunity still. There's still many, many millions of people. When the history of pandemic is written, boosters and alleged contraction of the 2 jabs will be seen as biggest BS. Morris, I do agree. I think a lot of the narratives that we're seeing will eventually be BS, as you say, but it will take some time. And right now, in the heat of the pandemic, the people who proclaim things, their voices rise to the top. Someone says, "Oh, this variant has 32 spike changes. That really worries me." And then it becomes, scientist is extremely worried about this variant, and it scares the hell out of people, and people don't know how to make a decision about it. So I think a lot of stories, yes, the contraction, which is the property of the immune response. I think the other thing is compressing the vaccine schedule between 3-4 weeks between doses initially. That'll be seen as a mistake. Many other things. And it will take some time to sort it out, and I hope I will point it out. Do you remember when, I will say, do you remember when? Well, it turns out that's BS. Yeah, that's a great point. No, SARS-1 also bound ACE2. DPP4 is MERS coronavirus. Yep. Oh, here's Mary M. I got the shoes yesterday at the incubator. And I will bring them to Amy Friday when I go up to Columbia. She thanks you. They're very nice shoes. I saw them. I showed them to Amy on our Zoom call today. Very nice. I found Spillover by David Quammen. Yeah, I have it right here on my bookshelf. It's a very good book. I like David very much. He has a very good view of things, and he's humble. He doesn't say he knows everything. He recognizes he knows little, and he needs to listen to scientists. Now, the problem is that not everyone he talks to is going to be right. But, a good writer has to suss that out And a good writer can sort it out. Not everybody can. A writer will often think, well, this person has worked on viruses for 20 years. They must know everything. But not everyone gets it. I'm not saying I get it all either. I'm wrong. But I think you have to be really careful about a lot of things. When a replicated virus leaves the cell and picks up an envelope from cell membrane on its way out, what happens to the spikes? Do they poke through the membrane envelope? So the virus picks up the membrane inside of the cell, right? From vesicles within the cell. Let me see if I can, while I'm talking to you, pull up a slide to show you, because a picture is worth 1,000 words. And so the virus picks up its spikes inside the cell, and then it moves to the surface in a vesicle. And then it's released at the cell surface. Let me see. I should have a picture. Here we go. Here we go. So let's share my screen. So, yeah, okay, it has missing fonts. Here's the virus being formed in the cell. It's in this Golgi intermediate compartment. Then the virus particle is complete inside the cell still in a smooth vesicle, and then the vesicle fuses with the plasma membrane, and then the virus particles are released. That's how coronaviruses are formed. So now, getting back to your question, what happens to the spikes? You saw in that picture, they are just inserted into the membrane. Let's go back. The spikes are inserted into the membrane in the ER, and then the piece of the ER moves over here, and the nucleocapsid pushes in, and that's how that acquires them. So hopefully that clarifies it for you. Not an Omicron question. My mom's coming back from China in 2 weeks, got 1 dose of Sinovac. Is there any problem with getting her 2 Moderna shots? No, there shouldn't be any problem. I'm surprised she can come in because it's not one of the, U.S. approved vaccines, but maybe they have relaxed that. But no, there's no problem getting two Modernas. Absolutely fine. I would just space them out a little more than 3-4 weeks, all right? As transmissibility is an intrinsic quality of a virus, how should faster passing through a population than another variant be described? So this is the problem where I clash with the epidemiologists, right? So I view transmissibility as an intrinsic property of a virus, and you can't measure that in people. And I'm going to actually make a video which explains it using the flu, avian influenza, H5N1 experiments in ferrets as an example, all right? Where they did incredibly carefully controlled experiments to say, here's a virus that doesn't transmit, here's one that does in ferrets, and here are the amino acid changes that mediate that. The problem with people is that we have not only the virus itself, but then we have what people do, and that influences transmission as well. So we use reproductive index as a measure of transmissibility, right? The problem is, there are 3 variables that go into that transmissibility, R naught. And 2 of them are what people do, and 1 of them is the virus. And so I would say you cannot tell much about the intrinsic properties in a pandemic when people are moving around and interacting in different ways. People are going to parties and bars, or they're not. You can't quantify that. I'm sorry. And so if I say transmissibility, I'm talking about the intrinsic property of a virus. And what's out there is actually fitness, the ability of a virus to displace other variants. And that's what says we have alpha, and then if delta comes, delta displaces alpha in 2 months. That's fitness. Yeah, delta spreads through the population, but that's because they're susceptible. If you still had alpha, it would do the same thing. It's no different. And they call that transmissibility, but it's actually spread in a population. They're not going to change the way they call it because of me. They say, well, who cares about what Racaniello thinks? Okay, that's fine. Not a problem. Moderna said they're applying to FDA for a full dose booster. Any time frame. Is it believed to be stronger? I don't know what their data are, okay? In my opinion, the booster data are weak to begin with. And Paul Offit will come on TWIV in a couple of weeks and tell you, because he did not vote for boosters for everyone. He'll explain it. He's on the vaccine committee, the ACIP. So a full dose, I doubt they have very much data on that at all, and I'm certainly not aware of it. All right, so this T index, right? It's the RT, or the RE, the effective R naught, because you can't do R naught after an outbreak has been on for a while, because you have population immunity, you have intervention. So then you have to do a estimated. And this is just what I'm saying. You can't make observations of infection numbers with time, rates, and say this virus is inherently more transmissible. Now, this professor may not be saying that. He may be just saying how effectively the virus is moving through the population. And that, I would argue, is a property of fitness. What are your thoughts about getting an mRNA booster given the new variant? Is it better to wait? Are boosters effective at all? Jennifer, I'm not going to tell you what to do because nobody's with me on boosters except Paul Offit, apparently, and a few other people. I contend that the data don't indicate that a booster is useful. And then people say, well, this is public health, so even if we don't have the data, just to be sure, you should give people boosters. And obviously, it's a recommendation at this point. It is not a requirement. You're fully vaccinated with 2 doses of Moderna, Pfizer. So it depends how old you are, if you have comorbidities. I think the variant itself does not warrant a booster. You make antibodies against all the variants that could possibly be out there and the ones we haven't seen yet. Yeah, you do have antibodies against all of them. I think it's really important for everybody to understand that. And nobody who talks to CNN or the damn New York Times says that. They just preach gloom and doom. For God's sakes, why? You can make any antibody. That's the beauty of your antibody generating system. And given the right vaccine, you can deal with any variant, and that's the point of the Binash Hatzianu paper that I talked about before. So your answer, if you want to feel safe, get a booster. It's fine. I haven't got a booster. I don't feel it's needed. I don't feel Omicron makes it any more needed than before. I certainly haven't seen any information that would say that. If I did, I would tell you. I'm not saying this to be contrary. I'm just going by the data. They would never, ever live stream this. They want to do their own thing and make a lot of money on it. Molnupiravir. Yes, the number I saw was 50% in the interim results. So maybe you're saying the latest results it's dropping. I don't know why it's so low. And the Pfizer protease inhibitor is much better. So maybe they're not treating the patients at the right time. Maybe they're treating them too late after. I just don't know the reason for that. It's an interesting observation. And I noted that initially as well. It's kind of disappointing. Some days 0 deaths from COVID in New York City. Is this more thanks to vaccination rates? It's vaccination. As Daniel said a couple of weeks ago, we have 4,000 new infections a day in New York, but 0 to 30 hospitalizations compared to almost a year ago, we would have over 400 hospitalizations. That's all vaccine. So a lot of these people are vaccinated. They're getting positive PCR tests. They're not very sick. They're not going in the hospital. The vaccines keep you out of the hospital. What more do you want? That's what it'll do against Omicron as well. It'll keep you out of the hospital. Do we yet know if infection after vaccination gives the same breadth of an immune response as infection prior to vaccination? I do not think the data support that yet. We are waiting. And I asked Daniel every week, and he said we're not there yet. So, and then the other question is, will a boost, say a third dose, do the same thing as a single dose after recovering from infection? That is, give you higher affinity antibodies, more broadly reactive antibodies that will recognize any variant that's out there. If those data are shown, then I'll get a booster. In my opinion, that's what justifies getting a booster. But we're not there yet. Would there have been some intermediate variants before Omicron with only fewer changes, right? Well, there could have been, but we're not sequencing every person on the planet, right? So those intermediate variants may not have done very well. Maybe they weren't very fit and they did infect some people, but we never picked them up because we only sequence a certain percentage of everybody who gets infected. So sure, it's absolutely possible. The UK is thinking that after the 2 doses of AstraZeneca and a booster, they want to give vulnerable an additional booster. Well, look, people get flu vaccines every year, right? But that's because the vaccines aren't very good, and especially older people, they don't have good immune memory to those vaccines. So older people get vaccinated every year. So it is one approach to preventing an infectious disease. And so, yeah, you could say we'll boost SARS-CoV-2 every year. Yeah, the 96-year-old immune system could handle it. I don't think that's a problem. I just think that the vaccines are better than that and that they don't need yearly boostings. But there is this impression that these variants demand it, and it's not the case at all. Their variants are still handled somewhat by antibodies and very much so by T cells. I am not caught up, but I heard you guys didn't like Fauci or dislike some of his messages. It's fine if people listen to him religiously. Everybody makes mistakes. Fauci has a very difficult job. Amy is very critical of him. I tend to be less critical because I think people need to listen to him. And if I'm criticizing him, then we're sending a mixed message. So I think he's fine. Nobody's 100% right. Can parts of the spike enter B or T cells via exosome leakage from an infected cell, then permeate the nucleus, impairing DNA repair? Sure, but why it wouldn't matter? It wouldn't matter at all because all the rearrangement for making antibodies has already happened many years before in the bone marrow, before you've even been infected. So it doesn't matter. Yeah, the isolation is terrible for South Africa. People don't make smart decisions about this isolation. Isolation doesn't matter. It's too late. Can you explain how we saw P61R displace P81H, but now Omicron is bringing 681H back? So it's all because there are multiple changes in the genome, and they interact with each other. They're what we call epistatic. That means the changes are interacting. And so you get one, and then some other thing changes in the genome, and so now you go back. Now, exactly what the function is that is being affected, nobody knows. But that is generally what's happening. And we see this from alpha to beta to gamma, delta, now Omicron. Some of these changes go back and forth, and some of the phenotypes go back and forth. So a beta had really high levels of resistance to antibodies, and delta less so. And delta displaced beta. So how to explain that? So it's not simple. And as I said, there's not a continuum of one variant to another. They are all adapting to different populations, and they're being selected for fitness in different populations initially. And so that's governing these changes going back and forth. It's really interesting. Gosh, we're not even 25% into the questions. All right, let's go here. Is there any part of the RBD, the receptor binding domain, that is resistant to mutation? There have been studies done a long time ago from Jesse Bloom's group, which showed that you could change a lot of the amino acids, but not all of them. They're obviously required for structural integrity of the RBD. Nobody knows what the membrane protein does. How is that? Well, people haven't studied it much. Not a lot of people work on coronaviruses, at least not before this pandemic. Should the Omicron sequences found in a country be expressed relative to how many genomes that were sequenced? They are. In this graph, in covariance.org, they express them as a fraction of the total genome sequence. And so in South Africa, it became, I think, 60-some% of the sequenced genomes over the last week or so. So they were getting a lot of those, for sure. I think proclamations of disease severity are extremely difficult to make. So, for example, we've seen people say alpha, beta, and delta get progressively more virulent, and then we have seen other people say, no, it's not true. So if someone says Omicron is milder, someone else will say it's more severe. And you cannot trust these observational studies in the midst of a pandemic that are done very quickly by just counting cases and applying statistical procedures. You have to do long-term laboratory studies as well to assess this. So I don't trust any study that says this variant is more or less virulent than another. If you've taken my virology course, you will learn that quantifying virulence in a laboratory is really difficult. So imagine in a human population where virtually nothing is controlled. How are you going to do that? Very tough. And most people don't know that. They think, we'll just count hospitalizations and account for sex and age and other variables, and that'll do it. No, it's not it. What distinguishes a T-cell epitope? It has a T on its forehead. No. They bind the T-cell receptor. You do assays. So B-cell epitopes bind antibodies or receptors, B-cell receptors on the surface of B-cells. Those are the antibodies bound to the membrane. And T-cell epitopes bind T-cell receptors on the surface of T-cells. And they're different. And B-cell epitopes change often because if you make a epitope resistant to your antibody, it's also going to be resistant to the next person's antibody. But if you make a peptide, a spike resistant to a T-cell, it doesn't matter for the next person because his T-cell or her T-cell receptor is very different. And so that's why, at least in acute infections that go quickly, T-cell variation does not have a big impact. Thank you, Galaxy, for your contribution. I'm 9 months after my second dose. Should I wait? You can, look, you're in the window where you can get it 6 months or later. If you feel nervous, get it. If you have comorbidities, you're young, I can't tell you what to do because people don't like my opinion. I'm 68. I'm not getting a booster. I'm healthy, but I am 68. Because I don't think it's needed. But you need to make your own decision. I can't do it for you. If you ask me, or does the data support boosters, I will say no, it doesn't. But people yell at me and they say, this is a public health issue. We have to be safe rather than sorry. Fine. If you want to be safe rather than sorry, go ahead and get it. Why don't public health docs believe ID and virology? They do. They do. It's not that they don't listen to me. For example, Daniel and I have great conversations. He understands what I say. I understand what he says. So I don't think it's correct. I think the problem is that people who speak to the press are not necessarily the ones who know what's going on. Why did the governor of New York need to declare a state of emergency? No reason. No reason whatsoever. She was given poor advice by people. Now, I would say that a year and a half ago, I was asked to be on the governor's panel to evaluate vaccines, and they never got back to me. I would be happy to sit on anyone's panel and tell them you don't need a state of emergency. That's ridiculous. So many people are vaccinated. That's all that matters. Oh, my gosh. Would you take a booster? No, I'm not taking a booster until I see the data. But that's me. I'm a rare person. You don't have to listen to me. All right, you're over 75, 2 Pfizers and a booster. How effective against severe disease and death? Oh, no, it's in the 90s. And if you've just had a booster, remember, you now have high antibody and T cell levels. So you're actually going to prevent any virus reproduction for a while, for, 4 to 6 to 8 months, depends on the person. So you're good, I think. And then as your antibody T cell levels contract, I think you're protected against severe disease and hospitalization in the 90th percentile. It's not 100%. Remember that, please. It's not 100%. And yes, people do not listen. The T cell epitopes, wrong person, have not changed. They haven't changed in any variant. They've changed a little bit, but not enough to make any difference. Because as I said, a changed T cell epitope doesn't matter for another person whose T cell receptor is different anyway. So Omicron is going to be recognized by T cells just like every other SARS-CoV-2. And those experiments haven't been done yet, but I'm sure Crotty and Sete are doing them, and give them a couple of weeks, and then I'll have them on Twitter to talk about it. The guy in Northern California with Omicron was vaxxed, has not reached 6 months for booster. So he had mild symptoms. And he said, I got to get a test. He got tested. He's positive. Why are we worried about this? The vaccines do not prevent infection. They prevent severe disease, hospitalization, and death. And I get letters saying, I'm sick of hearing you say that, Racaniello. Well, then stop asking stupid questions. If you don't get it, then ask questions until you do get it. And I'm not saying, Kitty, that you're asking a stupid question. I'm just telling you, people who yell at me for saying the same thing over and over again. It's true that T cells save your life. No, Delta is not, in my definition of transmissible, it is not more transmissible. It is more fit than Alpha and Beta and Gamma. And why it's more fit, we don't know. What does fitness mean? The ability to displace another variant. So all of a sudden you see Delta numbers go up and Beta numbers or Alpha numbers go down. That's fitness. It's not transmission as a fundamental viral principle. Almost everyone is using the word transmission wrongly. I use it to mean the intrinsic property of a virus that lets it go from one host to another, which, as I said earlier, is very difficult to determine. So you can't talk about that. Now they'll all use transmission. That's fine. It's the wrong word, and it's in every paper now, and it drives me crazy, and it's just going to shorten my life, but it's fine. It's not a problem. Do you think the cases in South Africa are being reported as milder due to the demographics, younger and healthier? Yes, that's a big problem. South Africa has a very young population. For sure, that's part of it. These are what we call confounding factors. You can't say the virus causes milder infections when the population you're looking at is different in every country. It's absurd. Is there a tipping point at which variants become strains? So a strain is a virus with some new property. And simply being more fit isn't enough. So it has to have we have to know what the reason for that improved fitness is. So why is Delta more fit? So we just did a paper on TWIV, which is dropping at midnight, that said, we looked at the reproduction of Alpha, Beta, and Delta in respiratory cells from people in culture in the lab. And we don't find any difference. So why is Delta more fit? It must be something that we haven't looked at. And that's what I'm talking about, figuring out what makes the virus more fit and makes it spread better in a population. Is it transmission? Well, then do a transmission experiment. Do it in animals in the lab, and with the caveats that it's just a lab animal experiment. Maybe it gives you some clues. There are very few of those out there. But don't make excuses that this is a pandemic, it's a public health emergency. That's BS. Just get vaccinated and everything is going to be okay. And we can't even do that, right? For many reasons. In countries that have vaccines, we can't do it, and we can't get it to countries who can't make vaccines. Are we essentially seeing the virus turn into endemic with similar antigenic drift as influenza? I don't think yet. It may be that that's the trajectory, that there is annual antigenic drift. We've had quite a bit of antigenic drift so far, right? And we're still using the same vaccine because the severe disease is still being prevented. With influenza, by the way, we see increase in severe disease, and that's when we change the vaccine. That's when we might want to change this one, or maybe a booster will prevent that. We don't know. That's what we have to sort out here. It's a good question. Ferrets are not people. That's right. But that's the best we can do because we can't make the right experiments in people. What makes me happy? Oh, everything. Everything makes. Even though I'm cranky, I'm very happy because I'm talking to 1,000 people. It's fun to interact with Amy. Every day is a good day for me, even when shit happens. I like talking about viruses. I like making podcasts and educating people. I like teaching. I like good food and red wine too. That makes me pretty happy, but you have the food and it's over, right? That's a good question, Miriam. As a general statement, a percent increase in transmissibility that can cause death is more deadly than the same percent increase in virulence as it's exponential. Are you making a statement? Oh, sorry, one person talking to another. My bad. Very, very sorry to intrude on your conversation. Is there any specific pressure or behaviors or actions have to evolve virus out of humans other than vaccines? Well, 1918 pandemic, the influenza pandemic, ended without a vaccine, right? So how did we do that? It just burned through the entire population, or most of it. And a lot of people died. So that's one way. You can just let the virus infect everyone and make a certain level of population immunity. But that's obviously not optimal. We don't want people to die. There are enough people who care about people not dying that we make vaccines and so forth. So, everyone could stay home, but we can't do that. That's obviously not practical whatsoever because there's some people who always have to get out. So the only alternative is to let the virus burn through the population. I read somewhere that people carry and spread the flu never become sick. Yeah, they have asymptomatic infections. It's also true for COVID, absolutely. 20% roughly of infections don't cause any symptoms. People walk around with it. Now, they don't walk around with it forever. They walk around with it for maybe 10 days. And then that's it. So if they walked around forever, that would be a disaster. But they don't do that with flu either. Are there common cold coronavirus variants? And are some of them variants of concern? So no, there are common cold coronavirus variants, antigenic variants. And that was only discovered in the past year by the Bloom Laboratory. We never thought that the common cold coronaviruses underwent antigenic variation. They are not variants of concern because few people care. They don't cause very severe disease for the most part, and so they're not of any concern, no. And they arise much less frequently, which is interesting because those viruses are infecting a lot of people. So. Have changes in human behavior over time resulted in variants appearing to differ in transmissibility? For sure. So I remember during the emergence of alpha, people say, it's more transmissible. We're seeing so many more infections. And then people say, well, actually, people have gotten out of lockdown and they're going to parties and bars and restaurants. And so that kind of behavior drives outbreaks, and it can fool you into thinking that it's actually the virus. I mean, there may be a component of the virus that makes it intrinsically more transmissible, right? And by now you guys should know what I mean by that. But it hasn't been determined what that is. And until you do, you shouldn't say the T word. But people will, and only I and a few others on Earth look at it in a specific way. Ron Fouchier is another one, besides Amy, who looks at it in a very specific way, the way it is meant to be used, transmissibility. This is an interesting comment by Steve. All this sequencing is like trying to forecast spread of a forest fire with a microscope. I think most of the sequence, we don't know what it means for sure. And as I said earlier, I think the outcome of the pandemic would be no different if we hadn't sequenced anything. Maybe the only thing is, we treat with single monoclonals and didn't work. Well, that would be because the virus became resistant, and you would pick that up clinically anyway. So Larry Brilliant said 4 to 5 years. Yeah, that's what Amy said too. 4 to 5 years. So what are we in year 2 now, right? January 2020, 2021. That's 1 year, 2 years in January. Probably 2 years more, yeah. Because most of the world is not vaccinated, right? And a country like the U.S., which should be more vaccinated, is not. So we still have outbreaks, although the hospitalization goes down. So that's good. The spikes were longer than 10 nanometers, would the aerodynamics be improved for transmission? So, Krista, I don't know, but this is exactly what I'm talking about when I say intrinsic properties of the virus that affect transmission. Nobody knows what they might be, and maybe, it could be the length of the spike. I have no idea. But that actually supports what I'm trying to say. 50% of the world has been affected, no end in sight. I don't know why you say no end is in sight. Of course, the end is in sight when you reach a certain percentage, which I always thought was 70%. Maybe now it's 80%. I don't know. But it depends what you look at. If you're looking at severe disease, we're already impacting that in New York City. And we can look at the percentage immunized there, and that could be a metric for the rest of the world. Whether flu changed in that epidemic is unknown. It will never be known. I think it's highly unlikely. I think herd immunity is a difficult one because to work, it needs to be homogeneous, and there are always pockets of people who don't have immunity. So, the 1918 flu pandemic, it was clearly herd immunity reached, and even if it was low, that was good enough to stop the transmission. I did say no variant has been proven to be more transmissible or virulent in my virological standards, not an epidemiological or hospital observation. They have not shown it. Perhaps in the next 5 years, the right experiments will be done to show it. In the meantime, it's SARS-CoV-2, and your chances of survival are massively improved by getting vaccinated, despite what variant has infected you. As Elena says, SARS-CoV-2 is SARS-CoV-2. Thank you very much. Thank you, HZoo, for your contribution. Really appreciate it. I'm a sequence jockey, but I agree, this is ebb and flow. Ebb and flow of genetic change. Yep. And yes, Lamont says, I think the sequencing will be great to mine in 10 years, but right now they're being abused. Great. Perfectly put, Lamont. And if I say this in other circles, they call me heretical. After being in Canada for 20 months, coming to the U.S., I'm convinced that human behavior, no masks and unvaccinated, increases transmission. Yeah, that does it. Now, it's clearly the movement of the virus, which you can call transmission. So how can you say that the Delta is more transmissible if you have all these human variables that are affecting it? That's why I look at a transmission as an inherent property of the virus, and then there's everything that people do, and I want to separate them. Because in the textbook that I write, I want to say, in this pandemic, these mutations arose that impacted the inherent transmissibility of the virus. I see zero data for that so far, and until I have it, the textbook Principles of Virology will not have that statement in it. I'm not just yelling, but that's what drives me, to get the textbook facts correct, because those in the end are what matter. It sounds weird, but getting the basic biology right is what I'm interested in. So Elizabeth says they should sequence. They shouldn't be guessing what the changes mean. I agree. I'm kind of saying that in tongue-in-cheek in a way, because I think it's being over-interpreted, as now we have seen many people saying. But yeah, it's useful in some ways, and we could look at it in 10 years, as someone has just said. All right, let's see if we can move through some of these. Do you think that children's doses should have been recommended to be spaced more than 3 weeks? So the main function of that 5 to 11-year-old study was to make sure that the vaccines were safe in children, no untoward side effects. So I don't think the spacing mattered. It was assumed that the immune responses are going to be similar to those in adults. Biden saying we won't lock down. In response, should I start buying tissue paper for lockdown? No, we're not going to lock down. Not happening again in this country. We had enough lockdown, and it's not necessary. I don't know. If Pfizer thinks they can make more bucks from an Omicron vaccine, they'll try it. Well, they have to get it past the FDA, right? But my point is, again, folks, I don't think it would have mattered if we hadn't sequenced anything. Wouldn't have made any difference. I'm not saying not to sequence, okay? You're misinterpreting my ranting. You know, if measles infects and spreads amongst vaccinated people. I keep telling people I think it does, so I can explain how this vaccine works, sorta. It does. It just doesn't cause measles in people. My 21-year-old had J&J. Since Moderna is cutting the booster dose, should she get it or Pfizer? 2 J&J? I think you're okay with 2 J&J, but if you have 1 J&J, you could get a second J&J, or either. It doesn't matter. The cut dose doesn't matter. It's not going to make that much of a difference, no. Thank you, Westfield 90, for your contribution. Really appreciate it. Chinese vaccine has not lost any efficacy against Omicron. No reformulation is not necessary. As I said, you already make antibodies against all the variants. If you're properly immunized, you will do that. So Campbell is just repeating what other people say. He has no way of interpreting information. And so people said it's less virulent, so he passed it on. More transmissible, he passed it on. There's no evidence for any of that, Thomas, as I've said. And so that's why I don't understand why a lot of people listen to Campbell, because he just regurgitates what others say without being able to make critical comments on it. But that's the internet. That's YouTube, right? Oh, the circles are lovely. I saw Amy's email. Very nice, Charlotte. Lovely. What's the best guess lately how long T-cell immunity will last? Well, Crotty has estimated at least 10 years. You're good. Vincent, in giving thanks to antibodies, that was just the name of our episode. Someone said that at doctors' offices they're testing binding antibodies, not neutralizing. is that because in order to test neutralizing, you'd need to introduce virus? So yes, you would have to have a pseudotype. You don't need SARS-CoV-2. You could use the spike in a VSV vector. But you'd need to do infectivity, whereas binding can be tested in a doctor's office by the lateral flow assays. Yeah. Yep. If somatic hypermutation broadens protection that much, why do we have variant-specific flu shots? Well, I think every virus is slightly different in the way that the antibody response covers it. And I also think that T cells play a big role for influenza virus, even though the correlates of protection is neutral antibodies against the hemagglutinin. I don't know the answer to that. It's a really good question. But I know that this happens with SARS-CoV-2. And that's why when you recover from infection and get a single vaccine dose, you get this incredible broadening because you're encouraging somatic hypermutation. How is the variant affected testing? It depends on the test. Many tests look for multiple targets, so it doesn't affect them at all. Some PCR tests will not register the variant, so you get a negative, which can also be epidemiologically useful. But all the tests can be modified to cover it eventually. Why random hexamers? Oh, it just was picked early on. That's better than shorter ones because the shorter ones are not specific enough. Well, actually, that's not true. A hexamer is not very specific either. A longer would be too specific. So a hexamer is the right size and it hybridizes better. Although you should ask Amy that. She would know better than I. Yeah, I said 70% or so population vaccinated creates herd immunity. So it is creating herd immunity against disease, right? We're seeing less and less disease. Most of the severe disease is seen in people who are not vaccinated. So in that sense, the herd immunity, is working. But we're still in a position where too many people are not vaccinated, and so a lot of disease in that group. They're not being protected because they're getting infected. The people who are vaccinated are not making disease, and that in a sense is herd immunity because they can be exposed to virus, right? And that virus doesn't transmit well from them, and it doesn't cause severe disease. Is there an increase in the chance of death in the following 12 months if you survive COVID? I haven't seen that number actually. I think it if it's been studied, I'm not aware of it, but I haven't seen it. No. Amy is a BS-free zone. That's the thing you guys have to get. So some of you don't like her personality, but no BS. If you ask her a question, she tells you what she thinks about it. And I think that's refreshing that you can have people who do that. I don't really give BS, although she says I do. I guess I try to finesse a little bit that she doesn't like. Is there anything about COVID you've heard recently that has surprised you? I guess the narrative around Omicron surprised me. I would think that people would understand the vaccines are working. Why would this variant be any different? Even if it has more changes, so what? There's no reason to think that the T cells are being run around. So that really surprises me, that you can have such an explosion of fear from a variant. Thank you, Nate, for your contribution. Really appreciate it. I'm pretty sure the vaccine has kept me from getting a cold sore for 8 months. I've never gone more than 2 months without one. Is it possible that the mRNA vaccine dampens? It's possible, but the effect would only last for a short time. It shouldn't be 8 months or so. I would suggest that you're less stressed because you're vaccinated and you understand that you're protected. Because as you know, stress reactivates herpes viruses. I don't think it's the mRNA vaccine, but it could be wrong. We'd need a study to do that. It's very interesting. Right after vaccination, you can have reproduction in the nasopharynx. Can a vax person theoretically infect their spouse or children if they're immune compromised? So right after vaccination, the antibody levels are the highest, so you're not likely to have an infection progress. It's 6 months later where the contraction of the antibodies would then permit infection. So yes, a vax person could bring virus home, and if an immune-compromised household member doesn't have any antibody or T cells against the virus, then that would be a problem, yes. So theoretically, because I don't know if the vaccinated person would be shedding enough virus to threaten them. And part of the equation is that only, 80% of transmissions are done by 20% of the people with the highest amount of virus in their nasopharyngeal secretions. And so I think that a vaccinated person is not likely to have those high levels of virus. So I'm not convinced that there is substantial transmission. There may be some, and so that's what you have to ask yourself, am I willing to risk that low level of transmission to an immunocompromised person? Thank you for your contribution. Really appreciate it. And thank you, Dan, for your contribution. Really appreciate it. You test your patients, but not all do. I've spoken to many doctors who say, especially in student health services, students come in, they flu-like symptoms in the winter, here, Tamiflu. I'm not going to bother testing you. Let's see here. Let's speed through because we are reaching 10 o'clock and it's an amazing number of people, more than we ever had. Thank you for coming. Come back next week. But let's see if we have any other burning tests. Why wouldn't you test someone vaccinated with COVID symptoms? I think from a medical viewpoint, you would want to see if you should treat them. That's what I said to Amy earlier. Yeah. But she disagreed because she said with flu, we don't do that. So that was the narrative. SARS-CoV-1 did not evolve itself to extinction. We contained it because most of the transmission occurred at the peak of disease, which over 50% of the people were then in the hospital. So it's easy to contain the infection in the hospital. Not a lot of community transmission, not a lot of asymptomatic infections to transmit in the community. So SARS-CoV-2 was just not good at putting all the right things together. And we were able to stop it. But certainly if we didn't intervene, it would have gone longer, for sure. Yes, so in part that's used to decide whether to, tell someone stay home for 10 days if they test positive. You're absolutely right about that. Even though I think the data for transmission from a vaccinated person are not strong, that is the logic behind that. Thank you, Lisa, for your contribution. Really appreciate it. Does whole genome sequencing give any insight into how the virus originated? Whole genome sequencing of people? I don't think so. I think it tells you about what makes some people have severe disease or not, right? But as far as origin, I wouldn't say it's silly. I think it's a good question. I may be missing something, actually. I don't bathe in coffee. I have 1 cup a day in the morning, and that's it for the rest of the day. And I don't know. Amy never drinks coffee during the day. Why would you even say this, 1890, that she shouldn't be had? Just keep it to yourself. That's a personal thing. It's bound to insult her, and we have enough insulting going on in the world, right? When can you go to Spain? You can go anytime. Probably you just have to follow their regulations. Go. It's not a problem. Vaccinated, wear a mask, you're all set. We have to test vaccinated patients if they work in healthcare. Exactly. There's another good reason. Doesn't Orphate defeat antigen presentation? That's the idea, Paul, but I'm not sure it's completely proven yet. But that would be the idea. But, it seems the T cells seem to be working in the case of SARS-CoV-2 vaccinated people. Yeah. All right, let's move to the end here and thank some people for donating because it's late. Sylvia, thank you so much. Any studies on genetic predisposition? That I answered before. Thank you, Sylvia, for your contribution. A lot of great questions. I'm sorry to pass them over. Bring them back next week. I guess these are the usual people. They'll be back next week. Thank you, Brian, for your support. Yes, you don't have to give on Tuesday. Any day is fine. And your donations tonight are all tax deductible, folks. Ryan, thank you so much for your contribution. Really appreciate it. How many times do you have to get COVID before your body can fight it off? Well, it depends because we have some people here who get it multiple times. But if you get it and you get vaccinated, then I think you have what we call super immunity, which is not a term that some people like. But those are the observations. I think we can have a fine conversation about Omicron 1890, even though we don't have. We can talk about what we do have. A lot of people have questions that need to be answered, so I don't agree that we can't have a conversation. We can't have a hysterical conversation, that's for sure. Do the mRNA vaccines provide a T-cell epitope response? Yes, they do. Thank you, Oak Ridge, for your contribution. Bring back your questions next week. They're all good questions. Hey, Eru, I forgot how to pronounce your name. Eurico, how are you? SARS-CoV-2 is present and infectious in 20% of children's tonsils undergoing tonsillectomy without COVID. So, Enrico, is that infectious virus? Probably not. It's PCR, right? What's the CT value? Yeah, a lot of questions. Get in touch. Thank you for your contribution, and it's good to hear from you. Enrico is my friend in Brazil who had me there twice to do TWIVs. Nice to see you on this live stream. Thank you, Mark, for your contribution. Your advice is to drink heavily. Well, then you can't be lucid on live streams and classes and podcasts. But certainly tonight I may have a drink afterwards, or I might just go to bed. Thank you, Richard, for your contribution. Really appreciate it. And lots of great questions. But with 1,000 people, it's hard to get to them all. Thanks you, Evan, for your contribution. I had an interview with a Boston Globe reporter. He just had fundamental questions about mutation. He said, why do viruses mutate? When will it stop? Everything mutates. It'll never stop. Why? Thank you, Josh, for your contribution. I'm Covishield vaccinated. Yes, you can get Covaxin. You can do these mixings. Absolutely. Was there a TWIV episode that talked about boosters? Yes, we've had a few, and we're having Paul Offit back, who's not a big fan of boosters, so listen to his episode when it comes out in a couple of weeks, for sure. Thank you, the original Ace of Spades. There is no evidence for original antigenic sin with respect to SARS-CoV-2. There is original antigenic sin with influenza, where whatever you're vaccinated with, you always make an antibody response to the first influenza virus you had ever seen in your life. And that can be a problem, right? Especially if that's antigenically different from what is currently circulating. And there's no evidence that happens with SARS-CoV-2. So these anti-vaxxers don't know what they're talking about. They pull words out from articles and they think it's impressive. Thank you, IMI. Yeah, if you do two thumbs, it basically negates the first. But thank you for your contribution. It's 1,100 viewers. Damn good for a science podcast. Will the A&V vaccine address the fast vaccination rate? Depends on how quickly they can make it. It has to get backed, right? It could, for sure. Thank you, Oxytropis, for your contribution. Because, as I said, your body can make antibodies against all the variants. And so you don't need an Omicron vaccine. You just need to have a good vaccine that exploits that diversity. Thank you, Arnaut. Really appreciate it. Ro, good to see you. Thank you, Marge, for your contribution. Thank you, Overjoyed. Really appreciate it. Time to start living outside this disease, not in it. Media hype is making us all nuts. I'm trying to counter the media hype here. In all ways, not just Omicron, but everything else. Virulence, transmission, boosters, etc. Is it useful to use spike dropout? I think until you have the proper PCR, it can be helpful. But remember, more than one variant is associated with spike dropout on PCR. So you have to make sure that the other variant is not in your area. If you're thinking it's Omicron because there's a dropout, it could be something else, right? Thank you, Sylvia, for your contribution. Powerful metaphor about the booster would help. I'm clear about the no need, but need more reasons against the why not. There's no reasons why not. You can get it. There's no harm in getting it. If you ask me what's the science, and a lot of people do, I'll just say there's no science in my opinion, but no negative. I think that helps you make your decision. Thank you, Sandy, for your contribution. It's just a scariant. Yeah, didn't we use that in a title some time ago? I like that. Variants are scariants. They're all scariants. Thank you for your contribution, Riri Med. Any recommendation of a great Italian restaurant in New York with great ossobuco? I have to ask Amy. There's one down on 20th Street, which is amazing. I can't remember the name of it. I will ask Amy and we'll tell you next time. Come back next week. Thank you again for your contribution. Has there ever been a toga virus pandemic? Not in modern times. Not in modern times, certainly. There have been big outbreaks of flaviviruses, but they're not toga viruses. Alpha viruses, big outbreaks, chikungunya, not toga, no. A lot of good questions. I hate to mass them all up. My gosh. Joe, thank you for your contribution. Don't worry about the variants of concern. Really bad name. Thank you, Mario. Incubator Coffee Fund for visiting moderators. Yeah, I should have coffee for visiting moderators like Vanity Nutrition who came today. You're welcome to visit the incubator. Just let me know. We're in New York City, right by Penn Station. You can watch us do the magic. Thank you, Ronnie, for your contribution. I'm glad you like us. Thank you, Gabriel, for your contribution. Lovely. Thank you, Gabriel. And Enrico, thank you. Infectious, isolated, and culture. And so these kids have no symptoms. And do you have any sense? Maybe it's just an asymptomatic infection, right? Which are more of in kids. Interesting stuff. Hope you publish it. Thank you, Laura, for your contribution. Really appreciate it. Toga virus is the name for, because the envelope looked like a toga. Yeah. Isn't that a good name? Thanks, everybody, for coming by. Thanks to the moderators tonight. A lot of work with all these people. We hit a record, over 1,100 people. 720 thumbs up is also a record. I hope I've addressed at least some of your questions. And if not, come back next week because we'll be here. Thanks, everybody. Stay safe and good night.